presidential addresses. So I'm going to talk about actually something that's maybe a little bit more technical, so there's some biological implications to this. A lot of what I'll talk about is sort of our personal experiences with working with traits that we were hoping would have maybe not a simple genetic basis, but at least some sort of candidates that we can go after with major effect. It turns out that's not the case. And there's a couple of reasons why. So, like many of you, I'm interested in adaptation, which means I'm interested in phenotypes, right? And the types of phenotypes that we work on relate to adaptation to variable environments. So things like the way that insects fit their life cycles to fit into seasonal life cycles, or the way that insects respond to extreme cold or extreme warm temperatures. And as it turns out, for better or for worse, those traits tend to be highly polygenic. And in fact, as we are accumulating more and more sequence data, what we're learning is that really many, even most traits that we're interested in, actually are polygenic in some way, shape, or form. And so these are all distributions of effect sizes for various human traits, human height being a classic example, but many human diseases as well, right? And most of the loci in these screens occur in the peaks, where effect sizes are very low. So there's positive effect sizes, negative effect sizes. Most loci have small effects. And there's very few loci out of the tails of these distributions, which is consistent with a polygenic model. And in fact, Pritchard has recently, and his group writ large, has extended this to what he calls the omnigenic model, which posits that essentially like every variant in the genome is making some small effect on the phenotype. And there's some pretty plausible evidence for that. So, given that that's the case, that sort of begs the question, what is the utility in doing a GWAS study if the effect sizes are so tiny? In other words, for people like us who are on NSF budgets and NIH budgets and have more limited sample sizes, we're working with non-model organisms, we can't do elaborate crosses, do we have a hope of identifying loci that will actually be informative? And that answer is actually surprisingly been yes in our system. I would expect it would have been more difficult. But so I'll show you a few pieces of data that suggest that's the case. And most of those data revolve around questions that are trying to test how these flies, Ragland are adapting to changes in their seasonality, rapid adaptation over just a few generations, just a handful. And so this is the sort of distastefully named apple magnet flies, which is quite a lovely little fly, at least in the adult stage. Many of you may know this because it's a textbook example of sympatric speciation, or speciation with chief of speciation. Sympatric speciation makes you angry for some people. So the flies are native to hawthorns, which are native to North America. They infest these hawthorn fruits. But sometime in the last 200 years or so, apples were introduced to North America, and the flies have established populations on those apples that remain genetically distinct over time, from generation to generation. And it's likely that there's a number of ecological axes that are being selected on to sort of wedge those populations apart. One of them that we have pretty well characterized is host odor preference, so what they like the smells of. Students in Jeff Vader's lab have done a really nice job of elaborating on that recently. My lab works primarily on seasonal phenology, and one of the important ways that these things are separated is in time. So the way this works is the fly is univolting, just one generation per year. It overwinters as a diapause pupae, and the adults emerge in late fall. And at a given sympatric site in the Midwest, apples screen about 3 weeks earlier than hawthorn flies, and the flies have evolved earlier emergence timing to track that. So it's a really obvious phenotypic target of selection. And so we've been doing a number of varieties of genome sequencing to try to figure out whether we can identify loci associated with that trait and infer things about population convergence. The two data sets I'll show you, well, several data sets of two flavors. RADseq, which is obviously reduced representation, so it's a little bit less than genome, and then some whole genome pooled resequencing, which covers the entire 1.2 gigabase genome with a little bit higher coverage. Okay, so the experimental design that we use is super simple. This is just a variant on what's called a classic bulk segregate analysis. The difference is that we're not crossing isogenic lines and looking at recombinants. We're actually just using outbred populations. So we bring individuals in, we rear them under common garden conditions, and we generate a distribution of emergence timing, and then we sample from the tails of that distribution. So the very earliest emerging and the latest emerging individuals. It's about 50 per bulk. And then we can estimate allele frequency differences, super simple, between those bulks, which is an estimate of the effect size of the locus on the And it turns out that you get a lot of bang for your buck. There's a lot of statistical power for relatively low coverage and relatively few individuals. The disadvantage being that you can't detect anything that's not sort of strongly additive. So that is our setup. The first thing that we've done with these data is just to confirm that the trait actually is polygenic. We have lots of sort of old phenotypic data to suggest that. But if we apply one of these sort of nifty Bayesian sparse linear mix models, which allow you to model simultaneously the effects of loci that sort of have measurable effect, that's in sparse terms in this model, versus random effects and sort of infinitesimal effects of loci that are affecting the trait that have very, very small effect. So this model tells us that about 63% of the phenotypic variation is explained by the genotype. So there are very clear genetic effects, but only about 16% of that variation is explained by the sparse effects, which are the major effects in the model. And in fact, I'm not showing you the error term here, but the credible interval for that value overlaps at 0. It's essentially negligible. So it suggests that the trait is in fact polygenic, as we guessed it would be. So can we detect associations? And it turns out that we can, and this is as evidenced by basically the repeatability. So originally we did this bulk segregated design on both Apple and Hawthorne flies from the St. Patrick's site because we wanted to know whether the genetic architecture of the trait was the same. But what that also gives us is basically a repeated GWAS study. We've done it twice in each of these geographic locations. And so if you take just a subset of loci, we can use permutation tests to ask which ones are sort of significantly associated with the phenotype. And we select a subset here. It's about 3,000 loci that are associated with the trait in both haw and apple. There's a very strong relationship. So those loci that are most strongly associated with the trait in the Hawthorne population are also highly associated with the trait in apple flies and in the same direction. So the correlation is relatively strong. If we use an even more restricted set of loci, and these are loci that are associated with the trait in both of those populations, we get an even stronger association. And that's really despite the fact that if you look at the scales, these are allele frequency differences, the effect sizes aren't huge, right? There's no fixed differences there, very few things that are approaching fixed differences. So, it appears to be repeatable. And not only that, we have done, for various reasons, the same study at different geographic sites and found essentially the same results. So these look a little bit different. These are whole genome pool resequencing data, which they don't display pretty on dot plot. So I have them in a heat map here. There's about 6,000 loci. This is at a different geographic site. This is Urbana, Illinois, the data I showed you from before. We're at Fenville, Michigan. Again, comparing haw occlusion association on the left side and apple occlusion association on the right side. The brightness just indicates the strength of the effect size for any given locus, and each row in that heat map is a different locus. So the punchline is that there's way more loci that are associated with the trait in both of these populations than you would expect by chance alone, and the effect sizes are again highly correlated. So green matches up with green, pink, well, I guess kind of purple here, matches up with purple, which means that there's a very strong correlation. It's the same loci basically oriented in the same direction, contributing to the trait. So, okay, so that's a lot of sort of technical mumbo jumbo to suggest that it is polygenic. We can actually find something interesting. So what can we do with it? We've done a couple of things. So this is one example. We have been able to use it because we can actually detect how loci are affecting the trait. We can use genetics to ask, to sort of confirm our hypotheses about what sorts of selection are driving these populations apart in nature. And so one of the first things that we did was simply to ask whether the loci that are most strongly associated with emergence timing in the field are also divergent among the host races. And that turns out to be the case. It also turns out that the directionality of this is completely confusing. My student talked about this yesterday through the magic of YouTube. You can go back in time and look at it if you're interested in it. Backlinking it talks a little weird. The other thing that we do, we're interested in sort of comparative physiology and insect development in my lab. And so what we want these SNPs for, in addition to asking these questions about host race divergence, we want to be able to leverage natural variation to help us learn something fundamental about how developmental processes work. And so, for example, these things, overwinter is dormant pupae. And the regulation of that dormancy that ultimately determines emergence timing happens during the winter. And so we've done things like collect brain transcriptomes across these sort of trajectories. We've identified genes that are progressively changing over time, that appear to be associated with the regulation of the trait, or that are differentially regulated among the host races. Without getting into the nitty-gritty of exactly how we're doing this, we're finding some really nice, strong statistical associations that cross-validate these genes with SNPs that are in cis regulatory positions near them. And so they're actually really useful. We don't have candidate genes. It's not genes. There's lots of genes, right? But it actually helps us to identify pathways that we think are important. So we drill down things like Wnt signaling and insulin signaling that we can now sort of pharmacologically inhibit through knockouts to tell us a little bit more about how this process is working in nature. Okay, so I'm kind of going to end on a cliffhanger here. In some ways, this is one of the most interesting results, but it's a relatively new result. We haven't quite wrapped our heads around exactly what it means. But it turns out that those GWAS results are highly repeatable within populations. So those urbana data are on the left here that I showed you before. We've done the same thing with pool-seq at another site. So this is a third geographic site, Grant, Michigan. Those are also repeatable between apple and hawthorn. Green matches with green, pink matches with pink for those two paired populations. But the loci that affect the trait are completely different, at least the ones that are associated in our analysis, between those two geographic sites. So the implications are that, one, we clearly have to be cautious about making inferences across the species range when we're doing GWAS in only a single population. The other is that, so Meredith Dolman and Jeff Fader's lab has published a couple of really nice papers recently about clinal variation of the species. So there's allelic variation that goes from south to north that we think would be associated with longitudinal trends in seasonality, which actually intersects with use patterns between the host races as well. But given this result, it's actually unclear whether we would predict declines at all. And when we see the declines, what's actually driving them, because those associations change so much over space as well. So again, I'm not sure exactly what to make of this yet, but this is sort of the next step in the research. We're digging into that right now. So I just want to quickly acknowledge, so Eddie is a co-author on this talk. She was a former postdoc of mine who's now at the University of Otago, and she worked on a bunch of this stuff. Tom Powell has also worked on these data as well. He's now at SUNY Binghamton. And then Hugh Robertson and Stuart Burlacher at IU are the progenitors of the genome that we use as well. Oh, and real quickly, if you're interested in bugs, seasonality, diapause, genetics, polygenics, my lab is recruiting for at least one PhD student and potentially a postdoc as well. So please come find me. Thanks.
So these SNPs that are different in different populations, are they like at least clustered at a genetic level, or are they really dispersed?
That's a good question. Yeah, so they are clustered across chromosomes. We have the assembly is not fantastic, but there's very clear sort of. So there's chromosome 1, 2, 3, 4, and then there's a fifth sort of sex chromosome. They're really on only two of those chromosomes, and that seems to be true for both of those populations. So you're right, it's not completely different, but it's also not completely